Hi everyone, I'm Laya Ekhlaspour, a pediatric endocrinologist at Stanford University, and happy to be here with you at DiabetesMine Innovation Days. Two weak points of automated insulin delivery systems are exercise and meals. In my work, I deal with meal challenges. In today's talk, I'll share with you my research that looked at how we can help algorithms identify meal composition from real-time CGM data. Patients using closed-loop systems understand very well that perfect glucose control will not happen just by an accurate carb count. And the complexity of their food matters, and it's just not about pre-meal insulin dosing or the amount of insulin. It's not just about the amount of carbohydrates, but the complexity and how much protein fat is in the meal also impact blood sugars after a meal. Dietary fat has been shown to impair insulin sensitivity and enhance hepatic glucose production, in addition to delaying gastric emptying through several mechanisms. In addition to dietary fat, also dietary protein has some glycemic effects, mainly through gluconeogenesis. Currently, automated insulin delivery systems handle simple meals pretty well, but when patients eat complex meals, and probably the most well-known one is pizza, we see that how these systems start to struggle. I will talk about two studies using a closed-loop system while looking at the effect of meal composition. I think these two studies are key trials in this field. And one of them was published by Howard Rupert in 2013 in a randomized crossover trial that looked at glucose excursions and insulin requirements in patients who were using a closed-loop system. So the patients were randomized to either a high-fat dinner or a standard meal. This is at 6 p.m. And here on top you can see the glucose trend, and here you see the insulin delivery. High-fat dinners required 42% more insulin than low-fat dinners. And the patients had also more hyperglycemia for about 5 to 10 hours following high-fat meals. In this study, they also reinforced that individual differences were significantly correlated with individual insulin requirements. While some closed-loop systems may be able to handle complex meals and address increased insulin requirements after high-fat meals, they help highlight the differences in time to glucose peak and give us some insight about optimized insulin dosing in these systems. This is a study that was published in 2018 and looked at high protein and high fat meals following breakfast. That basically high fat and high protein meal led to increased time to peak by about 40 minutes, and those patients required 39% more insulin. How do our patients handle it? Even if our patients can't count carb and do pre-meal boluses, we see that how they struggle with high-fat and high-protein meals. In patients who use open-loop systems, we recommend them to use an extended bolus, which is the programmed dose over a certain period of time. And a good example for the use of an extended bolus is for patients who eat a high-fat and high-protein meal, but also in patients who have gastroparesis, which is a common late complication of diabetes. So I do have a couple of examples of patients, like these are user experience of some of our patients who do use closed loop systems for their diabetes care. Here, this is a snapshot of a patient who used the Control IQ, and Control IQ is the only system that does have an extended bolus option, but which is a question that do closed loop system need to have an extended bolus option. But basically here he had like at 7 p.m. had high fat dinner. Does an extended bolus help with these high blood sugar that lasted for a few hours after the meal? Here I have a graph of a patient who use a Medtronic system that does not have an extended bolus, and he had dinner here. You see this late rise after pizza, and the same patient, the same meal with a lower set point. So even without an extended bolus, but with a lower set point, the system was able to handle his high blood sugar. So these instances tell us that not all the closed loop systems can handle high fat meals, but I think there has been no systematic study of patient experiences using closed loop system to inform us how to improve automated insulin delivery system to handle complex meals. So far, there's no algorithm that is able to make adjustments automatically. There's one algorithm that I would like to talk about very briefly, Loop, which is, as you know well, it's not an FDA approved system, but fairly popular, especially in the Bay Area. This algorithm incorporates dynamic carb absorption, and the default carb absorption time in Loop apps are average of the glycemic index. As you see here, like a lollipop for a low glycemic index, taco for a medium, and pizza for a high glycemic index food. So dynamic carb absorption model is able to model some of this variability and allow Loop to respond more reasonably. So Loop is the way to get to this, but we're not quite there. To find out how automated insulin delivery systems can be improved to handle complex food, I wanted to investigate the user experience in a more systematic way. So after a series of studies, in our closed loop studies, I consistently found that there is a difference between high fat and low fat meals. There's a difference in their glucose trends. So as you know, for closed loop trials, we usually start with inpatient studies and then we move to a transitional phase, which in California a lot of times we do these studies in Airbnb settings, which basically there's like supervised settings, but the patients are free to have any sort of activity that they want and any type of food that they want. So I decided to look at a larger data set in free living conditions, and the patients were free to choose their type of meal, but because it was supervised settings, so the research staff were collecting the meal composition data. So I looked at the data for 110 patients, a wide range from preschoolers to older adults, and I included just what I call clean meals basically. I excluded if there was any meal or snack in the time window that I was looking, if there was exercise, or if CGM data was missing more than 20%, and if there was correction given. And I classified meals into high carb, low carb, high fat, and low fat meals. So I had 1,885 meals and snack that I had to exclude most of it. So the analysis was conducted using 309 meals, 108 breakfasts, 55 lunches, and 146 dinners. What we found in our analysis that high-fat meals had a higher mean glucose rate of change. As you see here, this was more significant between 105 minutes to 115 minutes. And you see the red line and the purple line, with a less and shorter effect if we compare high carb meal with low carb meals. And you see here the purple and the blue line. So the fat was the dominant contributor to a higher and later rate of change. If we want to compare the high carb, low fat meals with low carb, high fat meal, and high carb, high fat, and low carb, high fat meals. And the strengths of this study was that we had a large set of data, and these are like real world data, and meal composition were collected by research staff, and so they were reliable. But obviously the limitation is that we didn't have a standardized meal, and we had to exclude many meals due to confounding factor. But what is the significance of this higher and late rate of change? I think we can use this higher and late rate of change to automatically increase controller aggressiveness to compensate for delayed meal absorption and insulin resistance following high-fat meals. High and late rate of change can and should be used to develop algorithms that can recognize high-fat meals and adjust the insulin delivery accordingly. I'm currently working with a group of engineers in Padova University, and we expect to finalize the analysis sometime next year. So in closing, I believe glucose rate of change will play a key role in developing the next generation of automated insulin delivery systems. And if algorithms can recognize that you ate a particular type of meal without you having to tell the algorithm and then adjust insulin accordingly, we will be a step closer to fully closed-loop systems. And I invite you to apply this concept to the systems that you are developing. And if you would like to hear more about my work, you can email me. Thank you for your time.